Okay. So, good. Briefly, a little bit of information for next week's paper. This is the paper. We're going into DNA synthesis. That's our topic. Okay. Something about DNA replication timing. Okay. So a little bit of background. We're talking about DNA replication. We have to show a picture of the cell cycle. When does DNA replication happen? At what point in the cell cycle? You go from the growth 1 phase, move into S, which is synthesis. That's when replication happens. And then you go through mitosis. Chromosomes segregate, you get 2 cells instead of 1, which is diagrammed in this fabulous figure. And so if you zoom in on replicating DNA, you might see something that looks like this. There are particular regions of chromosomes called origins of replication where replication originates. Cleverly named. And there's a lot of variation in how replication originates. Different species have different numbers of these places where replication starts. Different origins of replication have different efficiencies. That is, sometimes replication goes slowly from one origin but goes quickly from another origin. There's a lot of variation in how different organisms actually go about replicating their DNA. And this is usually the case, where you have multiple replication origins along a chromosome. Because if you just had one per chromosome, it would take forever to replicate a human chromosome, for example. Literally, like you wouldn't be able to grow. There wouldn't be enough cells able to be synthesized to make an organism. It would take too long just to replicate the DNA. So you get replication starting all along the chromosome, and the bubbles, replication bubbles, spread together until they fuse, and then you've got a whole replicated chromosome. So next week's paper, conservation of replication timing. It's part of the title. So they're interested in how these replication origins, what they call fire, or when a replication originates, it's also called firing of replication. So some origins of replication start synthesis early in the S phase, some wait later in S phase, which is weird because wouldn't you think that we want to start them all early in S phase? But that's not true. And the weirdest part, or at least the most surprising part to me, is that this is reproducible. So one origin replication, if you pick that one that's on that spot of that chromosome, it usually has the same characteristics from generation to generation. So if it's a late firing origin, if it always starts replication late, it usually does that. It's not just random. So there's something about these cells, there's something about maybe the DNA sequence of the origin itself, that part where replication starts, that dictates whether it's going to be an early replicating origin or a late replicating origin. If it's going to be high efficiency or low efficiency and so forth. So conservation of replication timing reveals global and local regulation of origin activity. So you know that these people are going to be studying when different origins of replication start synthesis. And they're trying to figure out, is it something local, that is about the DNA sequence of the origin, that dictates whether that origin is early activating or late activating, or is it something else, which they call global and local regulation. And they find both. So you should already be excited because they found everything. It's both local and global according to them. So when you read this paper, think about it in terms of the title. Another useful exercise sometimes. To ask yourself when you're done reading it, does the data really support the title or not? So So here's a different version of the cell cycle, and the reason that I'm using this figure is because it makes a useful point. Somehow these authors, and this is the technique that you need to understand, somehow the authors need to figure out at what point during the cell cycle each replication origin starts replicating. So how are they going to do that? We've got a bunch of different replication origins, and we need to measure when they start synthesizing DNA. So how much DNA is in a cell in the G1 phase? 100%. I like that, it's relative. If you're diploid, what's 100%?
2 copies.
2 copies, yeah. Which we usually call 2N. N being 1 set of chromosomes, 2 being 2 sets of chromosomes, diploid. Okay. What is the ploidy of the cell after synthesis? It's double. So it's 4 temporarily. But yes, right? After synthesis, you go from being diploid to being tetraploid, then you get cell division, you go back to being diploid. So it's normal, it's not bad, it's a normal part of cell division. Leave it at that. Okay. And then back here, you're diploid again. And everything starts over. Okay. If you're looking at a replication origin on the top of this chromosome during S phase, what's the DNA content of this cell? Is it still 2N or is it 4N? Where this arrow is pointing, the part that hasn't replicated yet, it's still 2N. But at the same point in time, on a different part of the chromosome, you're 4N. So the point is that you can measure during S phase at any point, you can figure out whether or not an origin has already replicated or part of a chromosome has replicated by asking, are there 2 copies of it in the cell or are there 4 copies? If there's 4 copies when you measure it, that means it's already replicated. If there's still 2 copies, it hasn't replicated yet. Okay? So that's the key insight into what they're going to do. Is at any point, you just ask, what's the DNA copy number? And to do this, it's always relative to the graphs that you're going to see for next week. It's always how much DNA is in this S phase cell relative to a G1 cell. So G1 is the standard. You know that that's 2 copies of a chromosome. If you see twice that, 4N, then you know that replication's occurred. In just a second. So this is just another way to look at cells in the cell cycle. And here's a little bit of modern technology for you. Has anybody used fluorescence-assisted cell sorting before? This is what these authors, you have?
I mean, I've seen I was working a lot of
that. Yeah, that me too. I've never done it, but I've seen somebody do it. That makes me an expert. So I'll show you how it works in just a second, just one image. But this is demonstrating this concept that you can measure something about DNA content. In this case, it's fluorescence. So you might put a fluorescent dye on some cells. And if they're diploid, they take up 2N amount of dye. They fluoresce 2N much. If that cell has replicated its DNA, then that cell takes up twice as much dye because there's twice as much DNA to stain. It fluoresces twice as bright, essentially. So that's what this x-axis is brightness. So what's happening is this FACS machine, fluorescence-assisted cell sorting machine, is measuring the amount of fluorescence coming out of individual cells. And this is just a histogram on the y-axis. So how many cells has this machine analyzed that have that amount of fluorescence? So we have a huge whopping number of cells. What is that? 3,600 plus 4,000 cells-ish, around there, that have that much fluorescence. And we have some smaller population, 600, what the hell is that? 900, so it's like 600 cells that have that much fluorescence. So what do you think these two peaks represent? What is the really tall peak on the left? Which stage of cell cycle are those? G1. Those are diploid. Or 2N. Which cells have gone through replication? The ones that have twice as much fluorescence. That's the 4N or G2 cells. They've already gone through synthesis. They're about ready to undergo cell division, mitosis. Okay. And it's not a coincidence that this peak is at 50 relative fluorescence units and this one's at about 100. You double the DNA content, you double the amount of fluorescence. So you go from 2N to 4N, you get, instead of 50 units of fluorescence, 100 units of fluorescence. And then all of the little peaks that are in here, what's going on in there? That's cells in various stages of replication. So if you want to figure out the profile of how different origins work, or when they're active, which cells do you want to look at? Do you want to look at the G1 peak, that little shoulder of S phase cells, or the G2 peak? Yeah, if you look at just G1 cells, they haven't replicated yet. If you look at G2, they're done. You want to look at all the cells in the middle.
The number of cells.
That peak, the height is, the number of cells.
Why is the problem decreasing the number?
Mostly because, when cells hit G2, they go through mitosis faster. Could be one explanation. It's a good question. Why are there so many G1s and so relatively few G2s? Yeah, I think that's the case. That G1, there's that resting phase where the cell doesn't do anything. G1, most cells are in G1 at any point in time. They zip through synthesis, they hit G2, as long as everything is okay in the cell, then they go through mitosis and then they're back to G1. So the fax machine, now this figure on the left, helps us actually collect, these scientists, actually collect that population of cells physically in a tube. So what you do is you take a bunch of cells, like shown here, mixture of cells. They don't have to be expressing GFP and CFP. Just assume that you've labeled all these cells, as I said, with a fluorescent dye that binds DNA. Those cells in a liquid shoot past a laser, which is the coolest part about the fax machine. And the laser reads the amount of fluorescence that's being produced by each of these cells. You shoot these cells in a single stream, single column, one cell at a time past a laser detector. Laser measures a few different things about the cell, the most important of which for this is fluorescence level. This is flow cytometry. Yeah, you use a flow cytometer to do FACS, fluorescence-assisted cell sorting. So what's down here then is, and this is the cool part, so computer reads the information that's coming out of the photomultiplier, detect how much fluorescence, and immediately applies a charge to that tube that the cells are shooting through, and puts a charge on the cell membranes faster than I can snap my fingers, instantaneously. And so when the laser detects a cell that's got a lot of fluorescence, it charges it so that the cell flies through a voltage field and bends a bit and flies into one tube that you've got waiting to collect cells. And if it detects no fluorescence, maybe it negatively charges that cell and it flies off to the other side into a different tube. It's crazy that this works. One of my old boss's husbands used to build flow cytometers. He has a flow cytometry. I have no idea who came up with this idea or who made it work, but it's genius if you can do it. And so then the idea is what you do is you tell the flow cytometer, I want to collect cells with that characteristic, fluorescence in between this threshold and that threshold. So you shoot thousands, tens of thousands, hundreds of thousands of cells through a flow cytometer in minutes. And it just goes left, right, center, 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 left, right, 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 right. And in an hour, you've got tons of different cells, all that meet your particular characteristics. In this case, cells that are in S phase, currently synthesizing their DNA.
Are those the ones in S phase?
Right, so in this case, yeah, what we're trying to do is collect these cells. So anything that's got fluorescence levels in between 2N and 4N.
So depending on the fluorescence level, it changes the.
Okay. So in a nutshell, if the DNA has already been replicated, you should sequence it twice as often. There's more of the template DNA or the actual chromosomes to be sequenced. Okay. So getting close to the end. These are the types of graphs you're going to see pretty much exclusively in this paper. A replication profile. On the x-axis, we're going to have, just like that previous figure with the deep sequencing, chromosome position. One end of the chromosome on the left, the other end of the chromosome on the right. So imagine the chromosome sequence on the x-axis. And the y-axis, what do you think is going to be there? We're just talking about deep sequencing and read depth. And what are we measuring to know whether or not something's replicated already?
So why should we read depth?
Yeah, it's related, exactly. It's related to read depth, copy number, which is the same thing, except it's relative. Copy number. Remember, what's the starting point for DNA content in the cell cycle? G1 is how many N? 2N. And S phase goes from 2N to 4N in that range. So what this relative copy number means is they're dividing whatever copy number that S phase cell is by the DNA content of a 2N cell. So this ranges from 1. So if you've got an S phase cell that just entered S phase, it's 2N. So its relative copy number is 1, not synthesized yet. The maximum then is 2. When an S phase cell completes synthesis, it's got 4N, or twice 2N. So this y-axis goes from 1 to 2. 1 is where cells start in S phase, 2 is where they end. Okay. So we can look at the red chromosome, and this is what a replication profile might look like if there's a single origin of replication. What does this mean? How do you interpret this bell-shaped curve?
So that middle point's going to be where half of it is doubled already.
Oh, actually, yeah, you're.
To go all the way up to 2, correct?
Yes, it should go all the way up to 2. Good point.
So
Right.
that middle part is right where it's finished with the S2 phase,
Right, so synthesis is complete,
and G2 is the downstream
and then
for mitosis.
the rest of the remember if there's one replication origin, replication starts there. That DNA content in the middle of the chromosome reaches 2 on this graph immediately, and then as replication proceeds, you go down in slope. And none of the all of this, remember this is a population of cells, thousands of cells that we're adding the information together to get this graph. So S phase is complete in all of the cells here at the peak. And replication has not even started to reach the ends of the chromosome. down here, right? All of these cells are still at one relative copy number, 2n still.
So is blue a mirror image then?
So blue is a different strain. So you could actually, I probably shouldn't have erased red initially, because you could see something like this. So red and blue are two different strains. So blue is strain 1, or whatever color that is. Red is strain 2. So what does it mean that this peak, if there's just one origin per chromosome again, which is not true, but for simplicity, what does that mean? Remember, this is not just one cell, it's a population of cells. So if that's at 1.5, say, that means that roughly half of the cells in the population have already replicated, the other half haven't yet, on average. So that might indicate an inefficient origin, where it's an origin of replication, yes, that's the highest point on that peak. It's where replication's starting, but maybe some of the time when replication starts, synthesis fails, or it goes slowly, or that origin just isn't used as frequently, for example, in this blue strain as it is in the red strain. A few different options. So that's why this question at the bottom that's been here all along, what kinds of factors could cause these sorts of differences between different strains of the same species, or between different species for that matter. So that's what these authors are going to be doing, looking at different strains, looking at replication profiles, seeing differences, and trying to figure out why are there these kinds of differences in replication profiles? What does it mean? Okay. Questions? It's kind of heavy, but, and again, this is not meant to be, it is kind of a lecture, but it's not meant to be a complete dissertation on this. It's just to get you started thinking about being able to interpret these for next week. I think we covered it. So this is the last little bit. And this is really what these authors are doing. So if we drew exactly those same figures, let's say this was one parent, the red parent strain, the blue parent strain. If you take a hybrid, like shown here in the upper right, an F1 hybrid, you mate the red parent with the blue parent, so you get a hybrid, F1. So let's say this is the parental strain, the P0 red strain, the P0 blue strain, just like we're drawn on the other screen. What's the replication profile of this hybrid going to look like? What do you think?
Theoretically in the middle, but wouldn't it be towards the higher?
So you're saying theoretically,
But you don't know if
it might look like the average of the parents.
But you don't know if maybe the one that's faster, maybe it tends to lean towards that one. It's replicating at a faster rate than the lower one.
So somewhere in between the two.
It's
Now
like
you're telling me somewhere in between the two curves.
It's like the whole car washing thing. Does one person necessarily slow down the other person if you split up washing the car? It's not always necessarily an in-between number.
I don't know if I know about the car washing thing. You might have to explain that to me. So that's one hypothesis, that the hybrid will be, as most hybrids are in a lot of phenotypes, the hybrids are intermediate between the parents. So there's that. There's another option. This F1 individual still has one red chromosome that has the red version of the red species, DNA version of those origin sequences. The blue chromosome has the blue species version of the origins. So, what does that mean? The hybrid might also look like both parents at the same time. It might have one chromosome that has the red version, and it might have a blue chromosome that replicates just like the blue parent. So it's not necessarily the case that there would be an intermediate phenotype. It could look like both parents at the same time. So what are we going to see? That's what we'll find out next week. So this is really the goal of the paper, the ultimate experiment that they do. You look at both parents individually, you look at the hybrid, and you find out how the hybrid replication acts.